Good afternoon. So today we're going to discuss the topic about DNA extraction techniques. So first of all, as we know, DNA is the most important evidence in a forensic aspect, as it play a vital role in an investigation. So what are the techniques that we can use for DNA extraction? So today we're going to learn about the techniques. So what are the techniques that we can use? Alright, the basic involved extraction techniques is tissue disruption, cellular and nuclear membrane disruption. So third is extraction DNA from nucleus by various techniques. So first we are going to discuss about the tissue disruption. Tissue disruption, as its name suggests, that tissue disruption. So by name only we can get the idea that we are talking about the disruption of tissue. So how we can do that? So tissue disruption can be done by proteinase K, alkali treatments, mechanical disruption and boiling. So what is proteinase K? As we already know that proteinase K so what, as we already know that it is an enzyme. So we are going to use an enzyme which will break a bond between the disulfide bond and bonds between the adenine and thymine. So let's talk about that. Proteinase K, as I already told you, it's a serine protease and was discovered in 1947 in the extraction of fungi. Proteinase K is able to digest hair, as keratin, we have keratin in hair, so it can digest. So its name given by this proteinase K. Proteinase K is the most commonly used enzyme for the tissue disruption. Proteinase K break down the, what it do? It break down the bonds. So it breaks down the disulfide bond and a diester bond, due to which the tissue are broken down into individual. So what we are doing? We are breaking down the tissues, right? With the help of proteinase, with the help of enzyme. So it's a technique. Certain alkali solutions such as detergent SDS along with a strong base as a sodium hydroxide is used. So to break the bonds we need some solution or some enzymes. So we use this. EDTA, as we all know, EDTA is a most important chelating agent. So what it do? It chelate the divalent cation, that is Mg2 and Ca2 plus. It break down these ions which are required for the functioning of DNA degrading enzyme DNAs. They also destabilize the DNA phosphate bond. What I am saying enzyme, we are breaking down the tissues. So what, with the help of enzymes and with the help of EDTA, what we are doing? We are breaking down, and we are what? We are just disturbing the phosphate backbone. What we are breaking the bonds, we are breaking the ions, we are breaking down the DNA structure. Okay. So mechanical disruption is usually used for the disruption of hard tissue such as bones and teeth. DNA, we can extract DNA from hairs, teeth, bone. So we need some from hard substance. We can extract DNA from teeth also. We can extract DNA from bones also. So for that, these are hard, right? We can't break them just like that. So we need something for that. So what we use? Enzyme we use, proteinase K. Alright. So liquid nitrogen which is stored at 176 degrees Celsius is used to grind the hard tissue using pestle and mortar. As we know, we use the technique pestle and mortar. We break down into small pieces. So cryogenic grinders are also being used for grinding bones. So what we all use? Cryogenic grinders. We can use this grinding pestle and mortars. And boiling method involves the boiling of the sample. Obviously, as its name suggests, boiling. So we will boil at the 75 degree Celsius, due to which the cells heated and disrupt. cell and nuclear membrane disruption. As I told you the three methods. So first method we already did, that is tissue disruption with the help of an enzyme, with the help of an grinding, with the help of Mg2 plus ion breaking down of ions. Now cell and nuclear membrane disruption. How we will do the cell membrane and the nuclear disruption. Cellular disruption involves of breaking down the cell wall and extracting DNA from the nucleus. We're going to extract the DNA from where? Nucleus, no. In cell we have nucleus. Nucleus contains DNA material. So we're going to extract that. Various salts and buffers are used for the cell and nuclear membrane disruption. So what salts and buffer are used? We're going to study. SDS and SLS are ionic detergents and are used along with a strong base such as sodium hydroxide. Tritone X and Tris HCl are used as both detergent and buffer. As I told you that we're going to use the buffer and salts. So what salts what buffer we can use? We can use HCl, we can use Tritone X. Alright. Sodium chloride. Sodium chloride, how much? 50 molar. So 50 molar was used in the oldest day for cellular disruption. Now we have many different ways which we can use. Okay, extraction of DNA from nucleus. Now how we gonna do that? We'll see that. Okay, so what are the techniques that we can use to extract the DNA from nucleus? These are the techniques. What are they? They are phenol chloroform extraction or organic extraction technique, differential extraction, silica based extraction, boiling and Kylix extraction, FT card extraction, extraction from plant tissue, that is called as CTAP method. Let's learn about each step. Alright, phenol chloroform, as its name suggests, we are going to use the phenol as well as the chloroform. So phenol chloroform extraction is a liquid extraction. Phenol is also in a liquid form. Chloroform is also in a liquid form. So both are the liquid. So it's a liquid extraction technique in a molecular biology for a separation of nucleic acid from protein and lipids. So how we gonna extract? So from protein and lipids we gonna extract the nuclei. Got it? Alright. Then next is aqueous samples. Lysis cell or homogenized tissues are mixed with equal volume of phenol and chloroform. So what we gonna do? We gonna take a same amount of phenol and a chloroform. Got it? Then after mixing, the mixture is centrifuged. After adding, for example, if I take 50 ml phenol and 50 ml chloroform, so what I'm gonna do? I'm gonna mix it. Got it? So after mixing, the mixture is centrifuged. I'm gonna do a centrifugation and two distinct phases are formed. Two layers gonna form because the phenol and chloroform mixture is immiscible with water. So they are not soluble in water. So a layer gonna form. Alright. Phenol only has a slightly higher density than water. Phenol की density हमारी water से ज़्यादा होती है, थोड़ी सी. Therefore it is mixed with chloroform. Water नहीं mix होगा phenol के साथ. Phenol की हमारी solubility water से ज़्यादा होती, तो chloroform और water जाके mix हो जाएंगे. So what will happen? Chloroform and phenol will mix together but with not water. Alright, then water, therefore it is mixed with chloroform to form a mixture with a much higher density than water. If we mix both the higher density substance, it's gonna make a mixture of a higher density than water. This will happen. Now the aqueous phase is on the top because it is less than, and the organic phase. Aqueous solution will be on the top because its density is less. Alright, the hydrophobic liquid will partition into the lower organic phase. The proteins will remain at the interface while the nucleic acid as well as other contaminants such as sugar, salt remain in the upper aqueous. So what will happen? Two layers will form. Aqueous solution, aqueous layer will form and a interface will form. So in aqueous solution our DNA salts will be in the upper layer. We'll found them in the upper layer. The upper aqueous phase is pipette out and care is taken to avoid pipetting any organic phase or material at the interface. So we don't need a aqueous phase. What we need is a interface because it will contain a DNA. So what we gonna do? We gonna remove the upper layer with the help of pipette. Okay. Then what we'll do? This procedure is often performed multiple times to increase the purity of DNA. We're gonna centrifuge it for like 3 to 4 times so that we can get a pure form of DNA. We will repeat this process 3 to 4 times. So what we gonna do? We gonna centrifuge it again to get a purity of the DNA. If the mixture is acidic, DNA will precipitate into the organic phase while RNA remain in the aqueous phase due to DNA being more readily neutralized than RNA. क्या होगा अब इसमें? हमारा मिक्सचर हमारे क्या है? एसिडिक है, तो हमारा डीएनए क्या करेगा? प्रेसिपिटेट हो जाएगा. किसके अंदर? ऑर्गेनिक फेज में. ऑर्गेनिक फेज में हमारा डीएनए प्रेसिपिटेट हो जाएगा. बट जो हमारा आरएनए है वो कहाँ रह जाएगा? एक्वस फॉर्म में रह जाएगा. हमने पढ़ा ना 2 लेयर बनेगी, एक्वस एंड इंटरफेस. हमें एक्वस लेयर से कोई काम नहीं, तो ये हमारा आरएनए जो है वो एक्वस फेज में रह जाएगा. ओके. Then this process yield a large double-stranded DNA that can be used in PCR or RFLP. Got it? So क्या होगा? हमने सेंट्रीफ्यूज किया, हमें प्योर डीएनए फॉर्म हो जाएगा, डबल हेलिक्स स्ट्रक्चर मिल जाएगा हमको. तो क्या होगा? The disadvantage of this technique in forensic use, since it's time consuming, as we're going to have to repeat the steps again for 4 to 5 times. So it's a time consuming process and we have to use chemicals which are hazardous, and because of this 2-step process involving transfer of reagent between tubes, it's at a greater risk of contamination. So what is the drawback of this phenol chloroform technique is that first it is time consuming. It's because we have to repeat the steps again and again. Second is we are using chemicals which are hazardous in nature. So that will be a drawback. And third is what? Because we are removing the upper layer, which increase the contamination. Alright, so see, this is a process. Alright, this is aqueous layer, this is interface layer, organic phase, this is interface. What we want to do? This is DNA palette or RNA palette. Phenol, we know pH is 4 and pH is 8. So how we gonna do? We gonna centrifuge this tube. We'll get a layer like this. Protein here will be in the interface. We'll get our DNA, and this is RNA. Got it? So DNA plus RNA will make in aqueous solution. Now we gonna learn about the differential techniques or extraction, differential extraction. What is differential extraction? So, modified version of organic extraction method. As I told you, what we are learning today is the DNA extraction method. So, it's a modified technique of organic extraction method, first described by Gill and Gusty. Alright. Process to isolate the male and female DNA from sexual assault evidentiary sample. In which crimes we can use this? Obviously we are using DNA as an evidence, and we can use like in which kind of cases? Rape cases, sexual assault cases, we isolate the DNA. Alright. From a single evidentiary sample, a female fraction containing the DNA from the victim epithelial cells and a male fraction containing the DNA from the sperm of an assailant are isolated. So what will happen if we receive a case of rape or something like that, something assault case, then what we gonna do? We gonna use a sample. What we gonna do? We gonna fragment a fraction. We gonna use a female fraction containing DNA of a victim and epithelial cell, and a male fraction containing the DNA from sperm of accused. Alright, are isolated. We gonna isolate their samples. Protocol, what gonna steps we gonna follow? What is the protocol? So we gonna learn about the protocol. The procedure involves preferentially breaking open the female epithelial cells with an incubation in an SDS or proteinase K mixture. So what we gonna do? We gonna break the epithelial cell of a victim, and with incubation we will incubate the sample with the help of SDS or proteinase K mixture. Sperm head remain intact during this incubation. We are gonna do the incubation, but the sperm head will be intact. We not gonna disturb the sperm head. The sperm heads are pelleted and the supernatant containing the female fraction is collected and saved. So what we gonna do? We just gonna focus on a female fraction. So what we gonna do? We not gonna touch the sperm head that are pelleted, and we gonna collect the supernatant as it will contain the female fraction, and we gonna use it for the DNA extraction. The sperm pellet is washed several times to remove any residual DNA from the victim. So we need to wash it, to different it, to remove the impurity, to remove the other things. Okay, the sperms are subsequently lysed by treatment with SDS or proteinase. So what I told you, that we gonna use what? Proteinase K we gonna use, SDS we gonna use, alright, and DTT, that is diethiothritol mixture. Alright, the DTT is required to break down the protein disulfide bridge that makes up the sperm head. So what we gonna do? We gonna use this mixture of these three, and we gonna break down the sperm head. The sperm are impervious to lysis without the addition of DTT. Both the male fraction and the female fraction are then extracted with the help of phenochloroform, and the DNA is precipitated with the ethanol. So with this technique we will what? We will differentiate between the male fraction, that is and female fraction, and after this we will wash the what? Pellet remove कर देंगे and any residue DNA from the victim. Okay, this is the diagram of differential extraction. So what happened? We will take the mixture. This is our sample. In this we have both male and female DNA. So what we gonna do is, we gonna mix this sample in the tube which contain EDTA, SDS and proteinase K buffer, and we gonna centrifuge at 37 degrees Celsius, and now what will happen? Sperm head will come down and this DNA will be formed on the layer. So we gonna remove the supernatant. We gonna remove the layer which is formed. So this is the female fraction. Okay, after we get the female fraction, what we gonna do? We gonna add DTT lysis sperm head. So sperm pellet will be removed and we'll get this sperm pellet will break down and it will form a, what it will form? Male fraction. Sir, male fraction. Now we gonna read about this. Differential extraction work well in most sexual assault cases. हम ये टेक्निक डीएनए एक्सट्रैक्ट करने के लिए कर रहे, किस टाइप के केसेस में? असॉल्ट, रेप केसेस. सो डिफरेंशियल एक्सट्रैक्शन technique works in well in the most sexual assault cases to separate male and female, because we gonna separate the sample, that is male and female fraction from one another. However, some perpetrators of sexual assault have had a vasectomy, in which case there is an absence of spermatozoa. So in some cases we can find that we don't have male sperm, right? So azoospermic semen, that is without sperm, okay, cannot be separated because it don't have a sperm. So how we can separate, right? So without sperm cell cannot be separate from the female fraction with differential extraction. So we can't differentiate, right? We can't extract male and female both sample. So in this case of azoospermic preparator, the use of Y chromosome specific marker permit male DNA profile to be deduced in the presence of X's. So now what? Now we have to at least do the differentiation. So what we gonna do? We gonna do this chromosomal specific marker. We gonna use Y chromosome, because male is Y chromosome, female is X chromosome. Now we need a male DNA extraction, right? We need a sperm to differentiate. So we gonna use Y chromosome specific markers. तो उससे क्या होगा? Then it will allow us, what it will do? It will deduce, right? Male DNA profile will deduce from female. Got it? In the presence of female DNA. Okay. Now we gonna discuss about silica based extraction technique. Alright. So what is silica based extraction technique is? Silica based, as its name suggests, we are going to use the silica. Silica based DNA extraction is also called solid phase extraction. So in this method, DNA is reversely absorbed to silica in presence of high concentration of chaotropic agent. So what we gonna use? We gonna use silica. So it is also known as solid phase extraction. So chaotropic agents are used to disrupt hydrogen bonding. So what will this? We gonna use the chaotropic agent. So what this chaotropic agents gonna do? It gonna disrupt the hydrogen bonding, affecting the tri-dimensional structure of protein. So ये क्या करेगा हमारा? ये हमारे हाइड्रोजन बॉन्डिंग को डिस्टर्ब कर देगा, डिस्टर्ब कर देगा. ठीक है. तो कॉमन chaotropic salts used for DNA extraction are guaninum hydrochloride and guanidium thiocyanate. Alright, so what we gonna do? We gonna use salts, chaotropic salts. Which two chaotropic salts we gonna use? So we gonna use guanidinium hydrochloride, that is GuHCl, and guanidinium thiocyanate, that is GuCN. तो हमारे GuSCN क्या करेगा? Is more potent chaotropic salt and facilitate silysis and DNA absorption. तो हमारे इसमें से more कौन सा ज़्यादा strong है? GuCSCN. So ये क्या करेगा? ये हमारे क्या करेगा? जो भी salts हैं तो ये हमारा सेल लाइसिस कर देगा एंड क्या करेगा? डीएनए अब्जॉर्प्शन कर लेगा. सेल लाइसिस करके डीएनए अब्जॉर्प्शन कर लेगा. When hydrogen bond removes the DNA, become negatively charged, hydrophobic and get absorbed in silica. अब हमने जब इसमें से सारे हाइड्रोजन बॉन्ड हटा दिए, हाइड्रोजन बॉन्ड क्या करता है? हमको हेल्प करता है ना. तो ये क्या करेगा? हमने जैसे हाइड्रोजन बॉन्ड सारे हटाए, ये क्या हो जाएंगे? नेगेटिव चार्ज हो जाएंगे और ये क्या हो जाएंगे? हाइड्रोफोबिक. हाइड्रोफोबिक मीन्स दे नॉट गोना डिसॉल्व इन वाटर. Before they were hydrophilic, but now they gonna become हाइड्रोफोबिक एंड गेट्स अब्सॉर्ब्ड ऑन द सिलिका. वाटर में तो होंगे नहीं, तो ये क्या करेंगे, सिलिका के ऊपर अब्सॉर्ब हो जाएंगे. ऑलराइट, what are the steps we gonna do for silica extraction? So first we gonna do cell lysis and protein digestion. So how we gonna do cell lysis? This is carried out by proteinase K. We gonna use enzyme. So what we gonna do? We gonna use proteinase K digestion. This result in release of DNA from cell. So when we gonna use proteinase, what will it will do? It will release the DNA from the cell. DNA absorption onto silica. Now second point is how we gonna take the DNA on silica. So it will adding pleiotropic reagent for absorption. As I told you earlier, we gonna use pleiotropic substances or salts. What it will help? It will help on the in the absorption of DNA on silica. Third is washing. First step what we did? We did cell lysis. After that what we did? We did silica absorption. Now what we gonna do is washing. Washing is done using solvent like ethanol base wash. This is done to remove pleiotropic agents and other contamination. Now we gonna wash. So what it will help? Why we will wash? We will wash it to remove the extra things which is there. So we gonna wash it and all the pleiotropic agents and other contamination will remove by this step. Alright, next step is elution of DNA. How we gonna do? Absorbed DNA can be eluted by rehydration. पहले हमने क्या करा, हाइड्रोफोबिक बना दिया. अब हम इनको क्या करा, वापस से हाइड्रेशन करा, हमने इनको रिहाइड्रेट करा. सो हमने क्या करा, absorbed DNA can be eluted by rehydration with aqueous salt solution. The eluted DNA is double stranded, can be used for sequencing. Now we got the DNA, double helical structure. Now what we gonna use it for? Sequencing. This is our process. Okay, lysis, we will do lysis, and then we will gonna do what? We gonna wash and use this nucleic acid protein complex. We gonna make it absorb on the silica. Now illusion of nucleic cross protein, हमने protein complex बनाया और हमारा यहाँ पे आ गया. अब हमने यहाँ पे इसको सिलिका पे absorb कराया, तो हमारे को बैंड्स आ गए. Last topic, and it is last technique, that is boiling or calyx method. Alright, boiling, extraction of DNA from boiling. So by name only we are getting the idea that what we gonna do, we gonna boil. Alright, this technique is also called the calyx extraction method, was introduced in 1990s to forensic DNA laboratories. So calyx 100 is an ion exchange. This is the solution. It is an ion exchange resin that is added as 5% solution. Got it? What it is composed, and what it is made of? It is composed of styrene divinyl benzene copolymer containing pair imino diacetate ions that act as chelating agents, chelating groups, and bind to metal ions such as magnesium. So what it will do? It will contain what? Chelating group. So it will gonna bond with it go and binds with what? Metal ions such as magnesium. By removing the magnesium from the reaction, nucleus are inactivated and the DNA is protected. Now what we gonna do? We gonna remove the ions. So what will happen? Nucleus are inactivated because there is no ion. So the DNA is protected. Okay, this is fast, cheap and effective method because we don't have to do more steps, right, of DNA extraction. This is the first step towards PCR. Now, and amplify your template DNA. What we gonna do? We gonna run polymeric chain reaction. We gonna do PCR and amplify your DNA template. What we got right? You must maintain excellent sterile technique. As we all know that we need to maintain a good, sterile condition for DNA. Alright? So, you must maintain excellent sterile technique to prevent the contamination of your DNA extraction. Always use a negative calyx control to evaluate your technique during the extraction phase. Okay, now what are the steps we gonna do? What all steps we gonna do? So washing, we gonna wash our this thing. Generally, this is done by adding 1 ml of sterile DI water, distilled water, to the sample. This aids in the removal of heme and other protein that inhibit PCR. So after washing, these things will be removed. Heme and proteins जो क्या करता है PCR को रोकता है, inhibit करता है. This can be done at room temperature or 37 degree Celsius. After the incubation, the sample is centrifuged. हमने क्या करा? Incubation किया. Incubation के बाद हमने क्या किया? Centrifuged किया and supernatant is removed and discard. So we don't need supernatant. What we need is a DNA. So we gonna remove the supernatant layer. Okay. So how we gonna do that? Alright. So what we gonna do? We gonna use 5% calyx super suspension is added to the tube and incubated. What we gonna do? We gonna use 5% of calyx and we gonna incubate at 56 degree Celsius. Incubation times can vary from 30 minutes to overnight depending upon the yes, this is very important thing to remember. We need to incubate so well, it doesn't matter if we need to do incubation. In some cases we need to incubate like for 10 to 15 times. In some samples we need to do incubation just for 5 to 6 times. So it depends on the sample. Alright, this step is used to lyse the cell. We gonna lyse the cell. What this calyx do? It's bind to Mg plus 2 ions which inactivate nucleus, which I taught you. Now we gonna do what we gonna do? We gonna add the enzyme, that is proteinase K. Proteinase K at this step and function to break down the protein. Now with the help of this proteinase K enzyme, it will break down the protein. The sample is vortexed and then boiled at 100. Okay, what we did? We washed first. Then what we did? We add the 5% calyx. Alright. Now after that we use the proteinase K to break down the protein. Now what we gonna do? We gonna boil the sample at 100 degree Celsius for 8 minutes. Okay. What we gonna do? We gonna boil the sample because it's a boiling technique. So we gonna boil the sample at 100 degree Celsius for 8 minutes. This causes cell membrane to re-rupture, destroy cellular protein and denatures the DNA to yield single-stranded DNA. Why we are boiling? We are boiling, right? Why we are doing that? Because when we boil, what will happen? Membranes are ruptured. सारी इधर उधर खराब हो जाएंगी, टूट जाएंगी, है ना? रप्चर्ड हो जाएंगी. सारे सेलुलर प्रोटीन्स जो है वो सब खत्म हो जाएंगे. प्रोटीन सारे उबल गए हमारे, सारी उसकी जो अच्छी वैल्यूज थी वो सब खत्म हो गई. वो प्रोटीन का कोई यूज नहीं रहा. एंड हमारा क्या करेगा? अब जो हमारा डबल हेलिक्स डीएनए जो हमारा डबल स्ट्रैंडेड है वो सिंगल स्ट्रैंडेड बन जाएगा. द ट्यूब कंटेनिंग द सैंपल and the calyx suspension is then centrifuged. अब क्या होगा? हमारा सिंगल स्ट्रैंडेड डीएनए आ गया तो हमारा डीएनए और कैलिक्स जो सस्पेंशन है वो क्या करेंगे? सेंट्रीफ्यूज करेंगे हम. ठीक है. The beads and the cellular debris are pelleted. अब क्या होगा? जो हमारे बीड्स हैं और जो सेलुलर डेब्रीज हैं वो क्या हो जाएंगी? पैलेट बन जाएंगी. सुपरनेटेंट में डीएनए आ जाएगा हमारा, सुपरनेट leaving the supernatant containing DNA that is used for quantitation and PCR. So now we gonna do PCR. Many laboratories procedures include a purification and concentration step using a centrifugal filters units such as micron, 100 or centricone. So this is an optional, this is not an mandatory step. So some people follow it, some don't. Alright, this is my calyx extraction process. What we did, we add water, we made it cells and calyx. We add 5% washing के बाद हमने क्या करा था, 5% calyx मिलाया था, 5% हमने calyx मिला दिया. फिर हमने क्या करा था, बॉईल करा था, बॉईल करके हमारे पास क्या हुआ, हमारे पास हमारी सब्सटेंस आ गया, ऊपर डीएनए आ गया सुपरनेटेंट में, हमारे यहाँ पे इनके डेब्रीज और वो सब कलेक्ट हो गए थे. फिर हमारा क्या हुआ, डीएनए हो गया. अब ये हमारे डेब्रीज सब प्रोटीन वगैरह डिसरप्ट हो गए थे, रप्चर्स हो गए थे, है ना. अब हमारे पास डीएनए आ गया. अब हमने क्या करा, इनको रिटेन किया और डीएनए सिंगल एक्सट्रैक्ट में आके डीएनए एक्सट्रैक्ट किया. The calyx extraction process denature double-stranded DNA and yield single. As I told you, what we will boil and we will get a single-stranded DNA. It will break down DNA into a single-stranded DNA. Care should be taken not to transfer any residual calyx beads with DNA extraction. Now you have to be very careful about with the DNA sample. You can't do anything. If once you mix the sample, it's of no use. So you have to be very careful with the DNA sample. So when you are taking the supernatant, you should be very careful that you should not mix the both residual calyx beads and the DNA. Now what, after with the DNA extraction to PCR reaction, as this can inhibit PCR like Mg. Now it obviously अब कुछ contamination होगा, हम PCR पे रन नहीं कर पाएंगे, तो हमारे क्या होगा, अब tag polymerase which is used in PCR, because we have some ions in the PCR, in the PCR, so it will react with them and it will polymerase chain को वो थोड़ा disturb कर देगा, हमें contamination आएगा. Okay, now what advantages and disadvantages, क्या है. Advantages बहुत क्विकली हो जाता है steps, single tube reaction, हमें बार बार change नहीं करना पड़ता है. Reaction which reduce the potential for laboratory and induce contamination and sample switching. कम हमें टेस्ट ट्यूब हमें एपेंडॉप एक ही यूज करना है, तो हमारा कंटेमिनेशन चांसेस क्या हो जाएंगे, रिड्यूस हो जाएंगे. नॉन टॉक्सिक है बिकॉज वी आर नॉट यूजिंग एनी केमिकल एज सच. कॉस्ट इफेक्टिव, वी कैन डू इट. व्हाट इज द डिसएडवांटेज? सो डिसएडवांटेज इज it will give us single stranded DNA, not a double stranded DNA. That is the biggest drawback of this technique. Yield single stranded DNA cannot be used in RF. We can only use in PCR. We cannot use it in a RFLP, that is restriction fragment length polymorphism. FTA paper. Now what is our next topic is? Our next topic is FTA card or FTA paper. So we gonna read about that. A unique matrix for the rapid preparation and ambient storage of DNA. So if you ever went to any FSL for internship or you ever come across, there is a one card, that is FTA card. What it is used for, in whenever you go for a crime scene, we gonna use a FTA card to store our DNA. So what we gonna do? We gonna take a sample, a drop of sample, and we put it on that FTA card and we store that for further DNA extraction. So a unique matrix for the rapid preparation and ambient storage of DNA from whole blood and other biological samples. So what we gonna do? We gonna use this card. What we gonna do? This kind of card we'll have. This is known as FT card. What we gonna do? We gonna take a sample, for example blood or any biological sample, we're going to put a drop of that sample in this and we will put, and after this from this card we will extract. Alright. Okay. What is the use of FT card? Kills blood-borne pathogens on contact, immobilize DNA within the matrix, protects DNA from degradation, allows for long-term storage at room temperature. अब मैंने क्या बोला कि हम उस कार्ड को रख देंगे, हम उस कार्ड को कितने भी टाइम तक रख सकते हैं, for 1 year also, for 2 years, for 5 months, for any time, like there will be no harm to that, it will be not contaminated. So it's a very useful technique. We can use it at a crime scene. Okay, FT paper, it's a unique mixture of strong buffer, protein, denatured, chelating agents and a UV absorbing free radical trap. The reagents are impregnated into the cellulose based filter matrix such as Whatman BFC 180 or 310 ET paper. So another approach to DNA extraction involves the use of FT card. Either we can use a technique like what I told you, FT paper or a FT card. Both are the same thing. Alright, then FT card only stood for Fitzico or Flinders technique agreement. Alright, FT paper is absorbent cellulose based paper. Alright, it's the same, we can store it at a room temperature and all that. And yes, as a result, DNA FT paper is at room temperature over a period of several years. As I told you that use of FT paper simply involves adding a sport. As I told you, we're gonna take a sample, we can put a drop on that paper and we're gonna store that. Now, a small punch of a paper is removed from a FT card blood stain and placed into a tube. Now, how we gonna extract the DNA from that? That is our motive, no? We're going to extract the DNA, then how we going to do? So, what we going to do? We're going to tear a small piece of that paper which containing a sample. Got it? It should be with sample like blood or whatever. We're going to take a small particle of that small piece of paper. We're going to cut it and we're going to put it in a appendix, or we're going to put it in a tube. So, a small punch of the paper is removed from a FT card blood stain and placed into a tube. For washing, we're not going to use a paper, no? So, we're going to wash it so that all the material which is there, it will come in a solution. The bound DNA can then purified by washing it with a FDA purification reagent. Now, we're going to use a purification reagent, that is Whatman, Clifton and Z. Alright, to remove heme and other inhibitors. So, what we can, why we are washing? So that we can remove all the inhibitors if it's present. Alright, the purification of the paper punch can be seen visually because of the paper is washed and the hemoglobin red color is removed with the supernatant. Now what we did, we're gonna remove the paper because what we need is a sample and we got it in the solution. So we're gonna remove the paper and we're gonna remove the supernatant layer. We don't need that. Alright, the clean punch is then added directly to the PCR reagent. Now we prepare our solution. Now we're gonna put it in a PCR. Alternately, some groups have performed Kilex extraction of the FT paper punch and used as supernatant in the PCR reaction. Alright, this is CTAB. What is CTAB method for DNA extraction? Let's see. Alright, CTAB stands for cetyl trimethylene ammonium bromide. Alright, plant DNA extraction, it's a plant DNA. This is our last topic, I guess. After this, we're gonna finish. Plant DNA extraction, how we gonna do? We can be use both freeze-dried leaves and a free. So, we're gonna need a DNA from a plant. So, what we're gonna do? We're gonna use a leaf that it can be a dried or it can be a fresh leaf. Method has been shown to give intact genomic DNA from plant. So, we're gonna, what it will? It will give us genomic DNA. To check the quality of the extracted DNA, a sample run on an agar se, what we gonna do? We gonna run the sample on an agarose gel. Stain with ethidium bromide and visualize under UV light. We're gonna put it on agarose gel. We're gonna put a dye that is ethylene bromide. Alright, and we will observe it under the UV light. CTAB is the detergent used to break open plant cell and solubilize its content. Material needed are C-type of a centrifuge, chloroform and isoamyl alcohol, RNase S, RNase A, isopropanol, cold 70 ethanol and PE buffer. So what is the process? Grind 350 mg of leaf. What we gonna do? We gonna grind the leaves. Alright, in mortar and pestle by adding 4 ml CTAB extract. So what we gonna do? We gonna grind the leaf. Alright, with the help we gonna add the CTAB extraction buffer in that. That is 4 ml. Transfer this into a clean centrifuge. What we gonna do? We gonna grind and we gonna add 4 ml CTAB buffer, and now we gonna transfer it to the centrifuge tube because we gonna do a centrifugation. Incubate the sample at 600 degrees Celsius for 60 minutes. Now what we gonna do? We will keep it for a incubation. Incubation at 60 degrees Celsius for 60 minutes. Alright. Allow this to cool down at room temperature for 5 minutes. What we did? We did incubation. Now what we gonna do? We gonna allow it to cool down because after incubation it become hot. So we gonna allow it for the cool for the 5 minutes. Now what after doing that? What we gonna do? We gonna add 5 ml of chloroform, isopropyl alcohol, that is 24 by 1 proportion. Alright, and mix for 5 minutes. After adding isopropyl alcohol and chloroform, we gonna mix it properly. Alright, then centrifuge the mixture. Now we gonna do a centrifugation. First we gonna do a incubation and then we gonna do a centrifugation. Centrifuge the mixture at 230 to 1300 rpm for 2 minutes at room temperature. Transfer the now after centrifugation we got a layer, supernatant layer. So transfer the supernatant into a fresh tube because we don't need the lower layer, we need a supernatant. So what we gonna do? We gonna transfer the supernatant into a new tube, and then in that we will add 25 microliter RNase. We gonna add 25 microliters RNase. Mix the sample and incubate. Again we have to incubate 30 minutes at room temperature. Now after incubation, what we gonna do? We gonna add isopropanol and mix. We gonna add isopropanol and now gonna mix properly evenly until the whitey fluffy DNA precipitate appears. Alright, हमने क्या करा? हमने 6 ml isopropyl बनाया, फिर हमने उसको मिक्स किया. ठीक है ना? उसके बाद हमने क्या किया? सेंट्रीफ्यूज किया. Centrifuge the sample at 2300 rpm for 5 minutes. Now what we gonna do? We will get a DNA at the bottom of the tube. So what we gonna do? Now we gonna discard the DNA, discard the supernatant. Sorry, we gonna discard the supernatant. Now what we gonna do? We gonna add 8 ml cold ethanol. What we gonna remove the upper layer, supernatant. Now what is left is the remaining part. What we gonna do? We gonna add the 8 ml of cold ethanol and ethanol mixture and mix कर देंगे हम उसको. ethanol मिला के उसको mix कर देंगे. Centrifuge at, वापस हमने उसको mix करने के बाद क्या किया? Centrifuge किया 5 मिनट ले. फिर हमने क्या करा? वापस हमने अपना जो भी ऊपर supernatant layer थी हमने उसको discard कर दी. Now what we gonna do? We gonna air dry the pellet. We got a pellet. Now what we gonna do? We gonna air dry the pellet for 10 to 15 minutes. Alright, because there will be particles of ethanol. So what we gonna? We leave it for 5 to 10 minutes to air dry. What evaporation will take place and all the ethanol will be removed. Now what after that? We have to add 1 ml TE buffer, alright, and resuspend the above pellet. हमने अपना TE buffer मिलाया, हमको वापस हमारी जो लेयर थी हमको वापस मिल गई. सी हमारा ये है, हमने इसको प्रेसिपिटेट किया, ऐसे हमने सी टाइप कॉम्प्लेक्स किया, फिर सेंट्रीफ्यूज किया, फिर हमने वापस प्रोटीन वगैरह हमको यहाँ पे निकली गई, हमें जैसे ही कॉम्प्लेक्स मिला है हमें यहाँ पे एक लेयर आ गई. फिर हमने रिसस्पेंशन किया पैलेट का NaCl में, हमने ऊपर की लेयर हटा दी सारी, सुपरनेटेंट को हमने डिस्कार्ड कर दिया, फिर हमारा इथेनॉल प्रेसिपिटेशन हुआ और फिर हमारा प्योर DNA हमने एक्सट्रैक्ट किया. So we are done with the DNA extraction technique. Thank you.